Hello, everybody. Uh, I'm very happy to be here. So I'm Domagoj Doresic. I'm from the University of Bonn in Germany in the Hasenau lab. And today I'm here to talk about our publication, so our proceedings publication here on efficient parameter estimation for ODE models of cellular processes using semi-quantitative data. So since this topic is focused on ODE models rather than machine learning, I'm going to start with a short comparison between these two approaches. So on one side, we have machine learning, which is focused on data-driven insights, and it has quite some positives. It is usually easy to implement, and it's, as we see on this conference, very versatile in its application. But in most cases, it is also very data hungry. And on the other side of the spectrum, we have mechanistic modeling, and mechanistic models don't have this problem. So they can be estimated with much less parameters. And this is due to the fact that we encode the mechanistic understanding directly into the model. That makes it also then highly interpretable. And if the model is calibrated well enough, then it can be used for in silico experiments, among other things. However, these models are usually pretty difficult to create. And in this talk specifically, I'm going to talk about one major mistake or one common mistake that is made when creating these models. So the main question that I'm going to answer today is how can we compare apples to oranges? I'm going to go in a little bit more detail on what these questions represent, actually. And then two more questions about the credibility of the proposed method, and lastly, about the performance of the method. So let's start with an introduction to ODE modeling and estimation. So let's suppose we have some kind of a model. So we have some reaction network of chemical species, two reactants, R1, R2, that react together to create some intermediary species, I, and then that species can then be transformed into a product P, a biochemical quantity that we're interested in and can measure. This model, for instance, has two parameters, K and theta, and the model can represent a number of things from cellular signaling and metabolism, so on and so on. With some assumption, this model can be then translated into a system of ordinary differential equations, which basically model the temporal evolution of the chemical species through time with some initial conditions. The model then usually comes together with some data. For instance, let's suppose that there's 4 measurements across 4 different time points that measure the biochemical quantity of interest, P. This data can then be used to estimate possibly unknown parameters of this model. So let's suppose that the unknown parameter of the model is theta, and we would like to estimate what the most probable value of this parameter is for the data that we have. So how do we do that? We're going to use a method called multi-start iterative optimization. How it works is that we start with an initial guess of the parameter theta, and we simulate the model forward with numerical simulators to get a simulation of the data that we can then compare to the simulation of the model that we can then compare to the data. And of course, what we want is that the simulation closely resembles the data. Of course, this is not a good outcome, not a good simulation. So what we want is to update the parameter to get possibly a better parameter value than we can simulate compared to the data. And we do this until we don't change the parameter value theta. However, we can sometimes end in some local minima. And of course, we did in this case. We don't know that always. So what we do usually is just we start a bunch of times. So that's the multi-start aspect of this. Usually for the models that I estimated in this talk, it's 1,000 starts. So we start another point. We again optimize, simulate, and then possibly get a better estimation, a better simulation that more closely resembles the data. Then we can analyze the results. Specifically, there is something called uncertainty analysis to see how certain we are in our estimates, and plausibility checks of how plausible possibly how biologically plausible our simulations are. These can then lead to an update of the model. For instance, we can choose to introduce an inhibition in the model and see how that works, or we can generate some new data if we see that we need the new data. Now, these apples and oranges represent that it's not always simple to compare the simulation to the data. It's more like comparing apples to oranges. What do I mean by this? So I'm going to show this with an example. It's an example of Foerster resonance energy transfer, so FRET measurements. So FRET is usually used to monitor biochemical activity of chemical species. So for instance, suppose that you have two chemical species. This is the green and the blue one, and they can also not react and they can react. When they react, we call them active. When they don't react, we call them inactive. And we want to monitor their activity, their connection, let's say. How do we do that? So there are these fluorophores that are connected to either ones. So the donor one, the donor fluorophore, and the acceptor fluorophore. Then excitation light is, there's some light that's emitted, excitation light, to the donor fluorophores. So this you see with these cyan little arrows. And what happens in FRET is that when the probe is active, the distance between the cyan and the yellow fluorophore is close enough that the excitation jumps from the donor to the acceptor. What then can happen is that the acceptor emits light that we can then see. So this is an indication of interaction between some chemical species. How do we measure this? So there's a method called ratiometric imaging. And what it does is that we basically try to see the fraction of intensity in the acceptor channel, so the basically interaction, and how much we put into the cells, so the donor channel. So we're looking at a fraction of intensities. It has been shown that this relationship this measurement is highly nonlinear. And also it can happen, or in most cases, these F parameters are generally unknown. So this, in summary, this measurement technique can have an unknown and nonlinear measurement mapping of the active probes to the intensity ratio. So this is one example. There are a bunch of other examples, for instance, Western blotting or fluorescent immunochemistry. And we can pose this in a general way. So we have some kind of a biochemical quantity of interest that we're actually interested in and would like to measure. And there is some kind of a measurement process that can possibly alter the data in an unknown and nonlinear way. The output is some measurement data. And this data we then call semi-quantitative data, because it is quantitative, it is numbers, but we cannot really use it directly. So there's an intermediate, there's a middle step of this, some kind of a measurement mapping. And this is what we're going to focus on. So we're going to focus on how can we model this general nonlinear measurement mapping that transforms our data. Or in other words, how can we compare apples to oranges? Let's go back to our example. So there's 3 general solutions to this modeling. The first one is, probably some people already guessed it, a parameterized mapping. So we have the expression already. So we can possibly just estimate the unknown parameters f and use the parameterization. However, we don't have this information always. We need that this requires some detailed and you have to be sure that this relationship is correct. And this knowledge is not always available. And an alternate method is to use some linear estimation. We just basically pull a line. But this is, of course, not too good when the true mapping is very nonlinear. And lastly, we have the method that we propose, spline estimation. I'm going to go into a little bit more detail on this. So we use piecewise linear splines to estimate these unknown nonlinear mappings. Why do we use them? Because they're simple to implement and to work with. But they're also, on the other side, flexible enough to fit most nonlinear shapes, let's say, depending on how many knots you have. However, it's not so simple. We had to introduce some improvements in the method to make it more efficient and to make it converge better. So firstly, we embed the estimation of the spline mapping in a hierarchical fashion. So we estimate the spline mapping in the inner problem of the hierarchical optimization problem. We calculate the gradients analytically, and then this allows us to use gradient-based optimizers for the optimization of the model. And we use spline regularization. Basically, what the idea behind this is to push the spline toward the linear line such that if there is some nonlinearity of the spline, that it comes from the data and not as some optimization artifact. So we're focusing on, we're trying to make it very sure that it's nonlinear if it is, right? These then resulted in large speedups and improved convergence. On this plot, you can see it's a little bit blurry, but you see that the number of converged starts per hour, which is the y-axis, is much higher for the hierarchical gradient-based approach that we use. And then, again, coming back to our example, we can use these three approaches to try to estimate the true mapping of the model. So we perform multi-start parameter estimation with 1,000 starts for each approach. And on the right-hand side, we have the results. Firstly, it's very obvious to see that the linear mapping is not sufficient. Right? It cannot approximate this nonlinearity. Secondly, the parameterized mapping is doing pretty well, and that's something that we would kind of guess that it should perform the best. But what's very nice to see is that the spline estimation is performing basically as well as the parameterized mapping. And the difference between these two is that the spline estimation doesn't have any prior information on what the nonlinear mapping should be. So now that we know how to compare apples to oranges, we're going to answer two more questions about the method, two most important questions of how credible is the proposed method, so how certain are we or how confident are we in the estimated spline? And secondly, how well does it perform on a benchmark of models? And specifically in these ODE estimation problems, we are most interested in how well are the mechanistic parameters estimated. So that's the initial theta parameter, if you remember from the introduction. So for this evaluation, we use 4 published models. They're coming from different areas. They have a varying number of state variables, parameters, observables, and data points. All of these quantities are somewhat mostly like understandable, except probably this third one I didn't introduce yet. So we call observables the quantity measured. So the number of observables corresponds to the number of different quantities measured, different proteins or whatever. Yeah. Then for each model, we chose and applied nonlinear measurement mappings to make it estimated. So we created the benchmark. And then, so this is an example for model 1. For model 1, it has 3 observables. We chose 3 different nonlinearities that we applied to the data. So for the first one, it's a saturating effect. For the second one, it's a sigmoid. And then for the last one, we censored the data. And this is what the model should recover, the method should recover. And it does. It's nice to see that it does. So this is the credibility study. So here we show that the method is credible, that the spline estimates are credible. For this, we sampled using MCMC sampling to get samples of the mechanistic parameters. And then for each vector of parameters, we computed the optimal spline. So we solved the inner problem in some sense. And this then creates the credibility interval around of our estimation. And what is very nice to see is that the true nonlinear mapping always lies inside this credibility interval. So the method is credible in that sense. But also what we were very happy to see is that the spline estimates are qualitatively similar to each other, right? So when you get one estimation result, just one like line, you can be fairly certain that that is the shape of the mapping. That's going to be important in slide 16, I think. So hold that in mind. But let's go to the performance study. So the last question. We're going to extend for the performance study, we're going to extend to models with a varying number of unknown measurement mappings, because we want to see how does it perform if it knows a couple or if it knows only one measurement mapping and so on. So we want to see how it does on models that have a varying number of these unknown measurement mappings. So for this, we took the models. For instance, for the model M1, we have 8 model variants. This corresponds to either of the mappings being known or unknown, right? So the first model variant is all unknown measurement mappings are known. And this is the gold standard, right? This is like the completely known model that is going to perform the best. And then we have the first is unknown, the second is unknown, the third is unknown, and so on, right? And we expect that the performance then decreases as we remove this prior information of what the measurement mappings are. And then we compare 3 approaches. Again, we have the linear estimation, so the competing approach, let's say. We have spline estimation, so our approach. And then for a reference to see, we wanted to see what happens if you just remove the data of the observable that has an unknown measurement mapping. So basically that means, okay, we're measuring a couple of things, and let's say a protein that we measure, we're kind of thinking, it might be nonlinear, let's just not use that data, that kind of an approach, right? So that's going to be a reference of, is the method even doing any good, or just it's not using the data in any positive way? And this is the result. So a couple of things to conclude. Firstly, we see obviously that the blue, so the gold standard, is always the best. That's what we expect. We also see that with the increasing number of unknown measurement mappings, the performance generally tends to be worse and worse. But the most important thing that for us is that our method is performing the best compared to other approaches. Specifically for model M1, it seems to be that knowing and not knowing measurement mappings and then using spline estimation is as if you knew them. So it's as good as if you knew them. This is a funny thing that we noticed, is that in some cases, it happens that if you just discard the data with the unknown measurement mappings, it is better than using a false method, so not a good approximation, which is the linear method. So it kind of shows that having a wrong assumption of measurement mapping can sometimes do more bad stuff than good stuff, right? Now, coming back to the credibility. So because our method is credible in the sense that it gives consistent shapes of the measurement mappings, it can also be used as a tool for uncovering nonlinear measurement mappings. So let's suppose that you have a model and you are not certain whether some data is completely quantitative, some observables, and that they might be semi-quantitative. So what we propose is that we can use the method. You can assume that they are semi-quantitative, and then estimate the measurement mapping and see what comes out. And then if identity line comes out, that means that you really have true data. That's the left side, right? That you can use it as it is, right? If you have an offset line that's scaled, that means that you have relative data and that you should use a method for relative data, and so on. So it can be used as a decision graph for what to do with the data that you have. So in summary, we have developed and implemented a spline-based method for the integration of semi-quantitative data, or also uncovering of nonlinear measurement mappings. So this is a first iteration of this. It is already implemented in PyPesto and can be used. So this is the link to the GitHub repository of our package PyPesto. It has a bunch of other tools for parameter estimation. This is a little plug. But for the method specifically, there are also some improvement avenues that we should and will consider. So there is, firstly, symbolic function identification at the end of the method would mean that you can parameterize the method automatically. Secondly, we could introduce a joint sensitivity analysis, which would make the method just more efficient, and that's something we show in the paper that it's possible to do. And lastly, splines do introduce, with like how many more nodes you have, more parameters you have. So it's a lot of parameters. And there might be better function classes with less parameters that can model similarly flexible measurement mapping. So we should probably explore, and we will explore alternative function classes. And with this, I would like to thank my advisors, Jan and Stefan, and of course, my whole group, and you for your attention. And these are the links for the PyPesto and the publication in the proceedings.
Thank you so much for a stimulating talk. So now I think we still have time for 1 to 2 questions. Anyone from the audience dying to ask a question? Yes.
Thank you very much for the nice talk. You were mentioning this different type of function class. Did you consider the Gaussian processes?
Yeah, we didn't yet, but that's an interesting idea that we should consider. We were thinking about beta distributions as well. They seem to have like high enough flexibility for low amount of parameters, but with this, it's we have this like step of analytical calculation of gradients that might be hard to reproduce. Also, I can imagine for Gaussians, it might be a difficulty, like to have the same efficiency. But yeah, it's something to consider.
Maybe we should discuss. Yeah.
Yeah.
Anyone else? 3? No. I wanted to say 3, 2, 1, but.
Hello, very nice work. I was wondering, so this implies that to use your method, you need to perform some kind of measurement to the calibrator spline for each species, for each measurement method of...
So we don't need a measurement. So we don't need a fully observed system, right? We need measurements only of some things. And usually in these ODE models, you measure only some things, right? And so I guess this is your question, whether the model should be fully observed or not. Or?
Well, no, the species for which you want to estimate the spline, you need to perform, an XY measure sampling to estimate the spline. Or did I get your method wrong?
So the method uses no. So it's not a separate thing. So it's integrated in the estimation of the model. So it just supposes that the measurements are semi-quantitative, that some kind of an estimation, like a measurement curve exists, and then it includes it in the estimation. So it's like a joint estimation of the model and the measurement mapping.